So our next presenter is Professor Steve Webb from Monash University. Dr. Steve Webb is an ICU specialist at Royal Perth Hospital, a professor of critical care research at Monash University, director of clinical trials at St. John of God Subiaco, and deputy chair of the Australian Clinical Trials Alliance. He has been an investigator on trials with an accumulated sample size of more than 50,000 patients. He's a named investigator on more than $115 million of competitive research funding and has published more than 180 manuscripts, including ones in NEJM, JAMA, and The Lancet. And he has a particular experience with Bayesian adaptive platform trials and other innovative designs, such as cluster crossover trials. And today he's going to share with us some trials on identified treatment for COVID. Welcome, Steve.
Thanks very much, Susan. I've taken myself out of the Royal Perth ICU this afternoon for a short period of time. I'm very sorry to have not been able to have made any of the rest of the meeting. So RemapCAP is a global Bayesian adaptive platform trial that actually emerged in response to the 2009 swine flu pandemic, in response to the recognition of the need for globally coordinated infrastructure to be able to conduct time-critical randomised controlled trials in patients admitted to an intensive care unit. I enjoyed, Jonathan, your recollections of the early days of the pandemic. I've had it pointed out to me several times that on January 7th, as the global leader of RemapCAP, an email to myself from the global project manager and the co-lead in New Zealand, which said, I hear there's reports of pneumonia in China, to which I responded, we've had these false alarms before, and no pandemic please in 2020, we're not quite ready. Well, the last 16 months since then have been like being on a hamster in a wheel. Fortunately, the workload is just starting to diminish a little bit. But RemapCAP existed as a trial for conventional bacterial or influenza-related community-acquired pneumonia that was always intended to be able to adapt in the event of a pandemic, and adapt is what we've been able to do. So before I start running through some of the results that have emerged from RemapCAP, I just want to explain the way that some of the results are presented. The traditional endpoint in ICU trials and the endpoint in RemapCAP for non-COVID patients is day 90 mortality. But we didn't think that it was appropriate in a fast-moving pandemic to have to wait 90 days to get the primary endpoint. And so what we've done is move to an endpoint that's available much more quickly and can feed back information to clinicians about optimal treatment. So what we've used is an ordinal scale, and we've discovered there's some wonderful properties associated with ordinal scales. That's a composite of death in hospital combined with the duration of organ failure support in an intensive care unit up until day 21. And that's the organ failure support component is still of public health importance, because in many locations at many times throughout the pandemic there's been substantial capacity constraints with respect to intensive care bed availability, and even without an improvement in mortality, getting patients better 3 or 4 days earlier and creating ICU beds has been important. Unusually, I just need to point out that a beneficial treatment effect in RemapCAP is an odds ratio greater than 1, which is the opposite direction to conventional ways of reporting odds ratios, so bear that in mind. And you'll notice that there's not a single p-value within any of the things that I'm going to present, which is a consequence of the use of Bayesian statistics, which merely report the probability that something is superior to an alternative. Over the last 16 months, we've been recruiting at a high rate and expanding RemapCAP, and I'll come to some of that logistic growth in time, but I just wanted to start by reporting some of the results that have emerged from RemapCAP. The wonderful, amazing recovery trial was the first trial to report the beneficial effects of corticosteroids, but we were evaluating corticosteroids as part of RemapCAP. We ceased that domain of REMAP-CAP as soon as the results of recovery became available, but we subsequently in September reported the results from REMAP-CAP showing a 93% probability of benefit associated with the use of corticosteroids. In the case of REMAP-CAP, we were evaluating hydrocortisone in a dose and duration that was very similar to the dexamethasone results that were obtained from recovery. So that was stopped before it had reached the statistical threshold that we have as a pre-specified threshold for stopping, but it still provided quite useful support to the beneficial effects of corticosteroids. Simultaneous with that, REMAP-CAP together with half a dozen or so other corticosteroid trials, all of which had stopped in response to the results of recovery, published a wonderful initiative, which is a prospective meta-analysis, multiple trials pooling their results to have a combined meta-analysis of the results of trials that are yet to be published. And so contributing to strong evidence of the quite substantial beneficial effect of corticosteroids in critically ill patients, and at the same time that was adopted by WHO in their international guidelines with recommendation for the use of corticosteroids. Probably a cheaply available therapy that saved countless lives during the pandemic, and whilst the vaccine effort has been truly extraordinary, we still have unmet need, particularly in low and middle income countries where this result in particular continues to save lives. We were also the first to report on the beneficial effects of interleukin-6 receptor antagonists, so tocilizumab and saralimumab, high probabilities there of superiority with odds ratios over 1, and this translates into about an 8% absolute improvement in mortality, so a number needed to treat of around 12 patients for 1 extra survivor. Monoclonal antibodies against interleukin-6 receptor antagonists have been widely used, adopted in international guidelines in the UK, in North America and in Europe, and have again contributed to saving lives throughout the pandemic. I'm going to report now a series of results that are not yet published, but have some of them have been published in preprint form. All of them have been publicly announced, but not yet subjected to peer review. So we became aware of other trials that were evaluating anticoagulation, prophylactic anticoagulation. It was recognised that both arterial and venous thrombotic complications were common in patients with severe COVID, and that these were occurring despite conventional DVT prophylaxis with low-dose Clexane or subcutaneous heparin. And so that led to the hypothesis that preemptive therapeutic dose anticoagulation might be beneficial. ATTACK is a Canadian Institute of Health Research funded trial that was recruiting in Canada, Mexico and Brazil. ACTIV-4 was an NIH funded trial recruiting in the United States as well as a couple of other countries. Rather than having 3 separate trials, each of which would take longer to reach a conclusion, we joined forces to have all 3 trials contributing to the same statistical model at Berry Consultants, who are the statistical consultants for REMAP-CAP. And so we hit a trigger for futility in patients admitted to an ICU with respect to therapeutic anticoagulation, and then stopped that in December, and then a month later hit a superiority threshold for patients who were ward initiated. So to just report those results more specifically, you can see there a consistent effect with an odds ratio of around 1.5 in several pre-specified strata with respect to D-dimer level. We'd had a priori hypothesis that treatment effect might be modified by D-dimer levels. But in severe patients, those who are admitted to an ICU and receiving organ failure support, an odds ratio under 0.8 and with credible intervals that actually indicate a high probability of harm. So interesting here to see heterogeneity of treatment effect for the same intervention depending on the state in which the treatment is initiated. But again, these results have been incorporated into international practice. I'll just skip over that, which reports the same things. We haven't quite submitted. We're about to submit our manuscript with respect to the effectiveness of convalescent plasma. So this is about 1,800 or 1,900 patients, all admitted to an intensive care unit, randomised to convalescent plasma or to no convalescent plasma. There was a huge amount of hype associated with the possible effectiveness of convalescent plasma. It had emergency use authorisation in the United States, and tens of thousands, if not hundreds of thousands of patients in the United States received convalescent plasma. Both our results and the results of recovery, which were published, I think, a day or two ago, recovery in the UK recruiting predominantly ward patients, very congruent results between the two trials, having REMAP-CAP having hit a futility threshold with an odds ratio that's very close to 1, indicating that this is not an effective treatment. We also evaluated some antivirals. This is also not published but submitted for publication currently. The recovery and solidarity trials provided early evidence of the lack of effectiveness of lopinavir/ritonavir, a repackaged HIV antiviral, as well as hydroxychloroquine, which had some quite famous protagonists. We stopped recruiting to hydroxychloroquine at the time of the solidarity and recovery results, and when we've had the opportunity to evaluate them, we were very grateful for having stopped at that point, because in the critically ill, hydroxychloroquine results in substantially higher mortality, an important finding. We continued to recruit to lopinavir/ritonavir despite the results from those other trials, because they'd mainly been recruiting less severely ill patients, and we thought that the question needed to be independently evaluated in ICU patients, and we hit a futility threshold for lopinavir/ritonavir in November or December, but have been slow to get that result published. If anything, the direction of treatment effect of lopinavir/ritonavir is towards harm, and so we're starting to be worried that there's a systematic issue associated with administration of antivirals in critically ill patients. Interestingly, one of the ongoing domains of REMAP-CAP with respect to influenza is an oseltamivir versus control trial, but that's still recruiting. So you might ask, how does a single trial provide answers with respect to so many different treatments? And a lot of this comes from the flexibility that's provided by Bayesian adaptive platform trials that use a statistical model that takes into account treatment effect across multiple different domains of treatment, including that many patients receive multiple randomisations within REMAP-CAP, which leaves us in a position to evaluate interactions between treatments which don't normally exist in conventional trial designs. So, for example, we've been able to include in the anticoagulation manuscript, which is under review at the moment, that randomisation to tocilizumab or saralimumab has no impact on the treatment effect of anticoagulation. And similarly, that with the interleukin-6 receptor antagonists, that the treatment effect of these agents is additive above and beyond that which is achieved with corticosteroid therapy. So this sort of type of gradually building up a matrix of understanding of the impact of different treatments and how they interact is one of the features of this type of design. So in 2019, REMAP-CAP was just a community-acquired pneumonia trial. And to just sort of elucidate this aspect of multifactoriality, if you wanted to evaluate different empiric antibiotics, the duration of macrolide therapy, and the effect of corticosteroids, in a traditional paradigm you'd need to do sequential clinical trials. Whereas in RemapCAP what we have is a system that allows patients to be randomised to 1, 2 or 3 of those treatments, and the independent and interacting effect of each treatment assignment within the platform is on the same primary endpoint is elucidated from the statistical models. This is the current diagram of the complexity of RemapCAP. We're now recruiting patients in the ward as well as in ICU, and we have capacity to generate different estimates of treatment effect depending on whether the patient's a ward patient or an ICU patient. And then you can see that there are multiple different domains there, most of which are still running and for which I have no idea the treatment effect, but it includes interventions like antiplatelet agents, vitamin C, statin therapy. So these are the domains in which we've had conclusions. You'll notice there that there's a new version of the anticoagulation domain, and that's because we're now seeking to answer the question for patients who arrive in the ICU, if they've been anticoagulated on the ward, should you continue the anticoagulation? And for patients who've arrived in the ICU not anticoagulated, should they be randomised to low-dose conventional DVT prophylaxis, or what has become standard care in many parts of the world, intermediate dose, in the knowledge that high-dose therapeutic anticoagulation is harmful. And we're doing that through 3 statistical models, which there's a pandemic model, an interpandemic model, which has got the old CAP interventions, and a single model for ventilation. But that square there, that's what RemapCAP was in January 2020, and everything else has been added over the last 16 months or so. And to give you an idea of what's happened, we modified the protocol to make it suitable for the pandemic. We started recruiting before the WHO actually declared a pandemic. We added the ward patients state in July. We've had the opening of multiple domains, and we continue to have multiple domains under construction, and we've had multiple domains cease either because of external information or because of information that the platform has generated. We've expanded very substantially. We were in about 13 countries in January last year. We're now in 22 countries and continuing to grow. And this is our recruitment as of yesterday on our website. We're in 318 sites in 22 countries, have evaluated or are currently evaluating 46 different treatments, and have recruited 6,500 patients with suspected or proven COVID and made 12,000 randomisations. So I really want to just acknowledge the extraordinary work that's been undertaken at the Australia-New Zealand Intensive Care Research Centre at Monash University, where I've got my academic appointment. It's got a long pedigree of having produced very high quality clinical trials, and it's been very much up to the game during COVID. Acknowledge our funders, NHMRC, who funded RemapCAP through a project grant about 4 or 5 years ago. But in particular, I really want to highlight the work of the support of the Minderoo Foundation, who've been a very generous supporter and in particular supported our expansion into low and middle income countries, and a great many partners. Thanks, Susan.
Thank you very much. I will allow one question. Lucky you, Steve.
Thank you very much indeed. And I'm going to escape, I'm afraid. I've got to go back to the floor.
Can we say thank you for that?